Hello. In this video, we're going to be looking at how the nucleic acid vaccines against SARS-CoV-2 induce both humoral and cell-mediated immunity in terms of preventing COVID-19. So as usual with these videos, start out by looking at your fundamental statements at the beginning. And these are the detailed learning objectives that you'll need to know as you progress through this soft chalk lesson. So our goal here is to look at how the current vaccines available against SARS-CoV-2, the Pfizer, the Moderna, and the Johnson & Johnson vaccines at the biological level induce both humoral and cell-mediated immunity. So let's start out talking a little bit about the two messenger RNA vaccines for SARS-CoV-2, the ones manufactured by Pfizer and by Moderna. Now, these are messenger RNA vaccines in that they contain strands of messenger RNA that are wrapped in lipid nanoparticles. And the messenger RNA codes for a piece of the spike protein of SARS-CoV-2. Now, you remember when we talked about the life cycle of SARS-CoV-2 in Unit 4, that the S protein is the viral protein that enables the virus to bind to host cell receptor molecule called ACE2 enzyme that are found on a variety of human cells, including those in the nose, the throat, the lungs, intestines, kidney, and heart. And although we're going to call this the spike protein, this codes for a portion of the spike protein. So it doesn't cause any harm in the person vaccinated, but it still functions as an antigen, which will then stimulate both a humoral response, that is the production of antibody molecules, and a cell-mediated response, the production of cytotoxic T lymphocytes that kill virus-infected cells. So let's review the absorption process here that we learned back in Unit 4. Remember, the SARS-CoV-2 virus has these spike proteins on the surface along with several other proteins, and it's a portion of the spike protein that fits to receptors on human cells, the receptor being the ACE2 enzyme, as we talked about back in Unit 4 in the SARS-CoV-2 life cycle. Now, the lipid nanoparticles that coat the messenger RNA, they protect the messenger RNA molecules from being broken down by enzymes in the body. And in addition to protecting the messenger RNA from degradation, they also help the messenger RNA enter antigen-presenting cells, muscle cells, and other cells that are affected by the vaccine. The dendritic cells are going to be critical in initiating the adaptive immune response. The one disadvantage, though, to the messenger RNA vaccines is that messenger RNA becomes degraded at higher temperatures. It's a fairly fragile molecule. And that's why the current Pfizer and Moderna messenger RNA vaccines have to be stored at very cold temperatures. And again, if you need to review the life cycle of SARS-CoV-2, there's a link here to that. And if you want to see some articles that compare the three common COVID-19 vaccines currently in use in the United States, you can click on this link. And of course, there's always this little self-quiz you can do. So let's move on. Now, again, we're looking at the biology behind how the vaccines function, how they stimulate humoral and adaptive immunity. So I've given you a little background information on adaptive immunity that we've already had in our current unit, Unit 6. And these vaccines actually provide a very nice way of summing up a lot of the information we've learned in Unit 6 related to adaptive immunity. So here's some of the information we've learned previously. I'm just going to briefly go through some of this as a reminder for you. First of all, of course, we learned early on in Unit 6 about the Mhc pathways. Now remember, these Mhc molecules that our body makes are what enable T lymphocytes to recognize epitopes of antigens so that we can discriminate self from non-self. T lymphocytes can't directly recognize antigens. They can only recognize them when displayed on MHC molecules. And there are two MHC pathways, Mhc1 and Mhc2. Now, as we learned early in this unit, Mhc1 molecules are made by all nucleated cells in the body. And these are designed to allow the body to recognize infected cells and tumor cells that develop in the body and destroy them with cytotoxic T lymphocytes, the cells that eventually bind to infected cells and tumor cells and kill them through a process called apoptosis or cell suicide. Now, these cytotoxic T lymphocytes or CTLs are effector cells derived from naive T8 lymphocytes. Remember, naive cells are those that had not yet reacted with an epitope of an antigen. Effector cells are the cells that actually go out and do something, in this case, cytotoxic T lymphocytes for T8 cells, T4 effector cells for T4 cells, and plasma cells for B lymphocytes. So what happens here with the MHC is that during viral replication, as well as the replication of intracellular bacteria or the replication of tumor cells, either viral, bacterial, or tumor proteins that are produced inside the cell, what we call endogenous antigens, or those inside the cell, get degraded into short peptides or epitopes by organelles called proteasomes. These peptide epitopes then attach to MHC1 molecules that all nucleated cells can make. And once the peptide epitopes are bound to MHC1 molecules, are transported to the surface of that cell, where they can be recognized by a complementary shaped T cell receptor and CD8 molecule found on the surface of both naive T8 lymphocytes and eventually cytotoxic T lymphocytes. So it's the Mhc1 pathway that allows our T8 lymphocytes to recognize epitopes of antigens and eventually differentiate into cytotoxic T lymphocytes that are designed to kill infected cells and tumor cells. Now, the Mhc2 pathway, Mhc2 molecules are made by antigen-presenting cells, such as dendritic cells, macrophages, and B lymphocytes. And these are the ones that enable T4 lymphocytes to recognize epitopes of antigens. So again, the Mhc1 allows T8 lymphocytes to recognize antigens. Mhc2 pathway allows T4 lymphocytes to recognize epitopes of antigens. Now, what happens in this case is that the antigens are engulfed and placed in a phagosome or endosome, which fuses with lysosomes, and proteases within the lysosomes then degrade the protein antigens, such as viral proteins, into short peptides. Here, the peptide epitopes can attach to Mhc2 molecules, which antigen-presenting cells make. They're transported to the surface of the antigen-presenting cell. Here, they can be recognized by the T cell receptor and CD4 molecules found on the surface of both naive T4 lymphocytes and effector T4 lymphocytes. And of course, the T4 lymphocytes, as we'll mention down below, regulate both humoral and cell-mediated immunity through the cytokines they produce. A little more about our antigen-presenting dendritic cells, since they'll be critical in what we're talking about today. Dendritic cells, as we learned, are derived from monocytes. And they're located under the surface of the epithelium of the skin and under the mucous membrane, such as in the respiratory tract, genitourinary tract, and gastrointestinal tract. They're also found throughout the body's lymphoid system, and they're in most solid organs. Now, once the dendritic cells capture antigens through pinno and phagocytosis, they detach from where they are initially located under the epithelium, enter near lymph vessels, and are carried to regional lymph nodes. And by the time the dendritic cells get to the lymph nodes, they've matured, and they now can serve as antigen-presenting cells because they can present these peptide epitopes to naive T8 and naive T4 lymphocytes. that are located and go through the T cell areas of the lymph nodes. Now remember, dendritic cells produce both Mhc1 molecules because they're nucleated cells, and these are what allow the dendritic cell to activate naive T8 lymphocytes. But being an antigen-presenting cell, they also produce Mhc2 molecules so they can present peptides to T4 lymphocytes. Our T4 lymphocytes, or T4 cells, are the ones that regulate the body's immune responses through the cytokines they produce. Now, the naive T4 cells, the ones that have not yet become activated, have to be activated by dendritic cells, as we just set up above. That allows them to proliferate into enough cells to mount an effective immune response and then differentiate into T4 effector cells. And these are the ones that are going to produce the cytokines that regulate immunity. And there's many different subpopulations of these T4 effector cells that produce different cytokines for different immunological functions, like the ones mentioned here, Th1, Th2, TFH, etc. T4 lymphocytes have a T cell receptor, or TCR, on their surface that, along with the CD4 molecules also found on T4 lymphocytes, can recognize peptides from protein antigens bound to Mhc2 molecules of antigen-presenting cells, such as our dendritic cells. Although macrophages and B lymphocytes also function as antigen-presenting cells, as we learned earlier. So the T cell receptor basically recognizes the peptide epitope, and the CD4 molecule on the T4 lymphocyte recognizes the Mhc2 molecule. And during their development, every T4 lymphocyte made undergoes a random gene splicing reaction, allowing it to make a unique T cell receptor that will fit a specific peptide epitope on a specific Mhc2 molecule that has a corresponding shape. T8 lymphocytes, or T8 cells, on the other hand, their primary role is to kill infected cells and tumor cells by a programmed cell suicide called apoptosis. And like naive T4 lymphocytes, naive T8 lymphocytes have to be activated by antigen-presenting dendritic cells. That allows them to proliferate into enough T8 lymphocytes to mount an effective immune response and eventually differentiate in T8 effector cells called cytotoxic T lymphocytes. And they then can kill the infected cells and tumor cells through apoptosis. Now, the T8 lymphocytes have a T cell receptor on their surface, and that, along with the CD8 molecules that T8 lymphocytes possess, are able to bind peptides from protein antigens bound to Mhc1 molecules. And again, the T cell receptor recognizes the peptide epitope. The CD8 molecule can recognize the Mhc1 molecule. And like T4 lymphocytes, during their development, every T8 lymphocyte undergoes a random gene splicing, enabling it to make a T cell receptor with a unique shape capable of binding a specific peptide on a specific Mhc1 molecule that has a corresponding shape. B lymphocytes, or B cells, are the ones responsible for humoral immunity, or the production of antibody molecules. And these antibodies are critical in removing extracellular microorganisms and toxins. During their development, like T4 and T8 cells, each B lymphocyte becomes genetically programmed through some random gene splicing to produce an antibody molecule with unique specific shape that will fit some epitope of some specific antigen. And molecules of this antibody are put on the surface of the B lymphocyte, where they function as B cell receptors. Now, in the case of most protein antigens, like the proteins that are being produced in the SARS CoV vaccine, the B lymphocytes have to interact with effector T4 lymphocytes, a subgroup called TFH cells. And these cells produce the cytokines that enable the activated B lymphocytes that have recognized an epitope with their B cell receptor to proliferate into enough cells to mount an effective immune response, synthesize and secrete antibodies, differentiate into antibody-secreting plasma cells, and allow the body to produce the different classes or isotypes of antibodies that are needed. So that's a brief summary of the key cells that are involved back from what we learned earlier in Unit 6. Now let's get into how these vaccines are working. And we're going to start out by looking at how the vaccine stimulates immunity in muscle cells at the injection site. So injection of the vaccine into the muscle triggers those muscle cells to produce antigenic SARS CoV-2 spike proteins at the site of injection. And these will stimulate both humoral immunity, the production of antibody molecules, and cell-mediated immunity, where cytotoxic T lymphocytes and cytokines are produced to fight the virus and kill virus-infected cells. So kind of look what happens here step by step, looking at figure 2 down below. We'll go back and forth between the steps. Now, first of all, the messenger RNA containing lipid nanoparticles at the injection site fuse with the plasma membrane of muscle cells. And as the lipid covering fuses with the membrane of the muscle cell, that empties the messenger RNA molecules into the cytosol of that cell. And we see that in figure 2, step 1 here. So here's our lipid nanoparticle, and inside are molecules of messenger RNA coding for the SARS CoV-2 spike protein. And that lipid nanoparticle fuses with the cell membrane of the muscle cell, and that dumps the messenger RNA into the cytosol of that cell. Once the viral messenger RNAs are released, they bind to host cell ribosomes, where they're translated into molecules of SARS CoV-2 spike protein, which we see in figure 2, step 2. So here's the messenger RNA that's released. Ribosomes bind to the viral messenger RNA, and they're translated into SARS CoV-2 spike proteins. Now, some of these spike proteins that are synthesized by the muscle cell go through organelles called proteasomes. Remember, that's what processes endogenous antigens for presentation to Mhc1 molecules. So as these spike proteins go through the proteasomes, they're degraded into a series of viral peptides, which we see in figure 2, step 3. So here's the spike proteins going through the proteasome, and that enzymatically chops them up into viral peptides. These viral peptides are then transported to the rough endoplasmic reticulum, where they bind to grooves of newly synthesized immunity molecules called Mhc1 molecules, which all nucleated cells in the body can make, including these muscle cells. We see that in figure 2, step 4. So once these viral peptides are generated, they will enter the endoplasmic reticulum, where they bind to grooves in Mhc1 molecules being made by the endoplasmic reticulum of that cell. Once the binding occurs, the endoplasmic reticulum then transports the MHC molecules with bound viral peptides to the Golgi apparatus, which in turn transports the MHC1 peptide complexes by the way of exocytic vesicles to the plasma membrane of the host cell. And here they become anchored in the membrane of the muscle cell. We see that in figure 2, steps 5 and 6. So here's the viral peptides attached to MHC1 through transport vesicles. These are transported from the endoplasmic reticulum to the Golgi. And then the Golgi then packages them in exocytic vesicles, not shown here. And eventually they become anchored in the plasma membrane of that cell. Now remember, once viral peptide is bound to MHC1 molecules, it can be recognized by cytotoxic T lymphocytes that have a T cell receptor and CD8 molecule that fit that peptide on that MHC1. So what that illustrates is that not only is the muscle cell producing protein antigen spike protein, but also that spike protein is being processed and is going to stimulate cell-mediated immunity in the same way that would happen if one of our cells, which actually was infected by living SARS CoV-2. So it's not just producing antibodies against the virus, but also the cell-mediated immunity. So we generate the cytokines, and the cytotoxic T lymphocytes need to kill the virus-infected cells. Now, some of the spike proteins that are generated inside the cell migrate to the surface of the cell where they become embedded. Others are secreted by the cell. And those that are secreted can then drain into regional lymph nodes near the injection site. And we see that in figure 2, step 7. So whereas some of the spike proteins go through the proteasome so they can be attached to Mhc1 molecules to stimulate a cell-mediated response with T8 lymphocytes, some of the spike proteins become embedded in the surface of the cell and others are secreted by the cell. Those on the surface can be recognized by the B cell receptor B lymphocytes, and those that are secreted can drain into regional lymph nodes where again B lymphocytes can recognize those spike proteins with their B cell receptor. So in terms of human immunity, these spike proteins that are put on the surface of the cell or secreted from the cell function as antigens. These can be recognized by the B cell receptor of naive B lymphocytes that have a B cell receptor that fit that spike protein. And we see that occurring in figure 4 down here. So here's kind of a generic virus, an enveloped virus. And here's a B lymphocyte that has a B cell receptor, which is the antibody the cell's programmed to make and is found on the surface of that naive B lymphocyte. And in this case, this particular B cell receptor fits this spike protein on this enveloped virus, and that would activate this B lymphocyte, which would eventually lead to the production of antibodies against that spike protein. So eventually those B lymphocytes are going to become activated, proliferate, and differentiate into antibody-secreting plasma cells. Now, in addition to this, some of the spike proteins draining into regional lymph nodes, some of the vaccine also drains into area lymph nodes near the injection site. And here it can be taken up by our next cell, the antigen-presenting dendritic cells found in the lymph nodes. Also, when vaccinated cells die, the cellular debris contains many more spike proteins and spike protein fragments, and these can also be taken up by antigen-presenting cells like dendritic cells. And eventually the messenger RNA from the vaccine is destroyed by the cell, so there's no permanent trace of the messenger RNA in the cell after a period of time. So that's basically what's happening in the muscle cells in order to begin stimulating the adaptive immune response. After fusion of the lipid nanoparticle and release of the messenger RNA, that is translated by host cell ribosomes into SARS CoV-2 spike proteins. Some of those go through proteasomes where they can be chopped up into peptides, enter the endoplasmic reticulum, and be bound to Mhc1 molecules, which once the peptides are bound are transported to the Golgi apparatus to exocytic vesicles and eventually put on the surface of the cell. And at this point, the peptide on Mhc1 can be recognized by T8 cells that have a T cell receptor and CD8 that fit that peptide on that Mhc1 for cell-mediated immunity. In addition, the spike proteins are put on the surface of the cell and secreted from the cell. And here they can be recognized by B lymphocytes to stimulate antibody production against the spike protein or humoral immunity. Now let's take a look at how the vaccine stimulates immunity in antigen-presenting dendritic cells. As we mentioned earlier, T lymphocytes can only recognize antigens when they're presented by antigen-presenting dendritic cells via Mhc1 and Mhc2 molecules. So as we've seen, the vaccine triggers the production of antigenic SARS-CoV-2 spike proteins by muscle cells at the site of injection. Some of that vaccine drains into regional lymph nodes where it can be taken up by our antigen-presenting dendritic cells in the lymph nodes. Also, when the vaccinated cells die, the debris contains many spike proteins and protein fragments. Those too can enter lymph nodes and be used or taken up by antigen-presenting dendritic cells. So in figure 5, we're going to look at what happens in the dendritic cells in the regional lymph nodes to stimulate humoral and cell-mediated immunity. So the messenger RNA containing lipid nanoparticles that have drained into regional lymph nodes from the injection site fuse with the plasma membrane of dendritic cells and empty their messenger RNA into the cytosol, as we see in figure 5, step 1. So again, like the muscle cells, the lipid nanoparticles will fuse with the plasma membrane of the dendritic cell and dump in the messenger RNA coding for the SARS-CoV-2 spike protein. Here, the viral messenger RNAs are translated by host cell ribosomes into molecules of SARS-CoV-2 spike protein, as we see in step 2 of figure 5. So here's the messenger RNA again being translated into spike proteins. And as in the muscle cells, some of those spike proteins are processed by proteasomes. Remember, dendritic cells have a nucleus, so they can make Mhc1 molecules and process them via proteasomes. Once the spike proteins are chopped up into viral peptides, they will enter the endoplasmic reticulum, as they did in the muscle cell, bind to Mhc1 molecules. The Mhc1 molecules with the bound viral peptide will then be transported to the Golgi apparatus, which will then, through exocytic vesicles, put them on the surface of the dendritic cell. And now we have viral peptide from SARS-CoV spike protein bound to Mhc1 molecule. And those bound to Mhc1 molecules can be recognized by naive T8 lymphocytes. To activate T8 lymphocytes that have a T cell receptor and CD8 that fit that peptide on that Mhc1. But also remember, our dendritic cell is an antigen presenting cell, and it can also make Mhc2 molecules. So the dendritic cells can also take up the synthesized spike protein that's drained into the lymph nodes, and they take this up by endocytosis, where it's engulfed and put in an endocytic vesicle. And then these spike proteins are degraded by lysosomal enzymes into viral peptides, as we see in figure 5, steps 7 and 8. So in addition to the vaccine delivering messenger RNA to the cell, the spike proteins produced by the muscle cells previously and have drained into lymph nodes can enter the dendritic cell by endocytosis, be put in an endocytic vesicle. After that fuses with lysosomes, the proteins in the spike protein are degraded into viral peptides. Meanwhile, the endoplasmic reticulum is also making Mhc2 molecules. Now, it makes these plugged up initially so that the peptides here don't bind to Mhc2. They only bind to Mhc1. And these Mhc2 molecules that are temporarily plugged are transported to the Golgi. The Golgi transports them to the endosome. And here along the way, they become unplugged. And now these viral peptides from the spike protein are also attached to Mhc2 molecules. And through exocytosis, they're put on the surface of the dendritic cell as well. So here's Mhc2 molecules with bound viral peptide. And that's the one that's going to activate naive T4 lymphocytes for cytokine production. And here's the peptide bound to Mhc1 molecule, which is going to activate naive T8 lymphocytes so they can become cytotoxic T lymphocytes. So the net result of this is that as a result of the vaccination, naive T8 lymphocytes are going to be activated, and they will differentiate into cytotoxic T lymphocytes that can then kill cells infected with SARS-CoV-2, displaying SARS-CoV-2 viral peptides. In addition, it's going to activate naive T4 lymphocytes who recognize the peptide on Mhc2, and they're going to differentiate into T4 helper lymphocytes or T4 effector cells that are going to regulate immunity through the cytokines they produce. Now, also, of course, some of the spike proteins, as we saw in the muscle cell, migrate to the surface of the cell where they become embedded, and others are secreted by the cell. We see that in figure 5, step 7, wrong way. So some of these spike proteins get embedded, some can be secreted. And of course, then these antigens are what can be recognized by B lymphocytes, naive B lymphocytes, via their B cell receptor, and they will eventually become activated. And after interacting with T4 helper cells, will proliferate and differentiate into antibody-secreting plasma cells. So again, we're seeing a strong humoral immunity response, the production of antibodies against the spike protein, and a strong cell-mediated response where we activate naive T8 lymphocytes and naive T4 lymphocytes to eventually produce cytotoxic T lymphocytes and T4 effector cells. So here's kind of a summary of our antigen presenting cell. Our dendritic cell, remember, is going to attach different viral peptides to Mhc1 molecule because antigen presenting cells have a nucleus, so they can make Mhc1. And when peptide is bound to Mhc1, it can be recognized by a naive T8 lymphocyte who uses its T cell receptor and CD8 molecules to fit that peptide on that Mhc1. And that allows the antigen presenting dendritic cell to activate the naive T8 lymphocyte so that it can rapidly proliferate to give you enough cells to mount an effective immune response. And eventually those cells will differentiate into cytotoxic T lymphocytes that will bind to virus-infected cells displaying peptide on Mhc1 and kill those infected cells through apoptosis or cell suicide. We'll see how that works down below. But the antigen-presenting dendritic cell can also make Mhc2 molecules because it is an antigen-presenting cell. And so when peptide is bound to Mhc2, it can be recognized by the T cell receptor CD4 molecules of naive T4 lymphocytes that have a corresponding shape. And then the antigen presenting cell will activate that naive T4 lymphocyte, enabling it to proliferate into enough T4 lymphocytes to mount an effective immune response. And those cells will then differentiate into the proper T4 effector cells that will regulate immunity through the cytokines they produce. And as always, we have some self-quizzes along the way you can do. Now let's talk a little bit about the Johnson & Johnson vaccine. It's also referred to as an adenovirus vaccine, but it actually contains a DNA gene coding for the SARS-CoV-2 spike protein, just like the messenger RNA does in the Pfizer and Moderna messenger RNA vaccines. But unlike the messenger RNA vaccines that Pfizer and Moderna produce, remember those use lipid nanoparticles to deliver the messenger RNA that code for spike protein or S protein. The Johnson & Johnson vaccine has an added DNA gene coding for the spike protein SARS-CoV-2, which is found inside another virus called adenovirus 26. So the delivery system here is not a lipid nanoparticle, but a harmless adenovirus. Now, adenoviruses are a group of viruses that typically cause cold symptoms, but adenovirus 26 has been modified so the virus can still enter the human cell, but once it enters, it can't replicate. It can't make any more viruses. It can't cause any illness. And what it's doing then is delivering that gene coding for SARS CoV-2 spike protein. So after injection, the adenoviruses, and these are naked viruses that lack an envelope, they adsorb to receptors on the surface of human cells by means of some protein fibers that are part of the capsid that covers the genome of the virus. The virus enters the host cell by endocytosis and is placed in an endosome. And following acidification of the endosomal membrane is lysed. The viral nucleocapsid is transported by microtubules to the nuclear membrane of the host cell, which would be a muscle cell or dendritic cell. Here, the viral particle disassembles and the DNA genome that was in the adenovirus enters the nucleus of the host cell through nuclear pores. And this is shown in figure 7. So there's the harmless adenovirus 26. It's been modified so that it doesn't have the genes it needs to replicate. It can't replicate, it can cause harm, but it can still enter the cell because it still has the adsorption fibers on its surface. So that adsorbs to host cell receptors. The virus is taken in by endocytosis and put in an endosome. Eventually, after acidification, the endosomal membrane is degraded, and the nucleocapsid follows microtubules down to the nucleus where it disassembles, and the viral DNA enters the nucleus of the host cell. And in that viral DNA is a gene that codes for SARS CoV-2 spike proteins. So the viral DNA is transcribed into messenger RNA, which leaves the nucleus and attaches to host cell ribosomes, where, like the messenger RNA vaccine we discussed earlier, gets translated into spike proteins. So the means of entry is different, but everything else after that is the same immunologically. So once the gene for the spike protein is read and transcribed into messenger RNA and translated into spike protein by host cell ribosomes, these newly synthesized spike proteins stimulate humoral and cell-mediated immunity in muscle cells and dendritic cells the same way they do when they're produced as a result of the messenger RNA vaccines. So you can look at the illustrations above for how immunity is induced in muscle cells and how immunity is induced in dendritic cells. It would work the same way here once the virus has entered the cell. One advantage of the Johnson & Johnson vaccine is it seems to require only a single injection rather than 2 that are needed with Pfizer and Moderna, the messenger RNA vaccines. And because DNA is more stable, this can be stored at refrigerated temperatures rather than the ultra-cold temperatures needed for the messenger RNA vaccines. Which brings us how all of this gets rid of the virus, should you become infected. So again, this stimulates both antibody-mediated and cell-mediated immunity. So remember, these antigenic spike proteins that the muscle cells and dendritic cells are producing cause the immune system to produce antibodies that bind to the spike protein of SARS CoV-2. And we'll see that that's going to eventually block absorption down here. It's also going to activate T4 lymphocytes and T8 lymphocytes to fight the infection. T4 lymphocytes to produce cytokines needed to regulate and amplify all of this. T8 lymphocytes to become cytotoxic T lymphocytes that kill virus-infected cells. So again, the vaccine is inducing both humoral immunity, the production of antibodies, and cell-mediated immunity, the production of cytotoxic T lymphocytes and regulatory cytokines. So let's first of all look at B lymphocytes producing antibody molecules specific to the SARS-CoV-2 spike protein. Now, as we said, these B lymphocytes have B cell receptors on their surface, which are the actual antibody that cell has been programmed to make. Every B lymphocyte becomes programmed to make a specific B cell receptor antibody that fits a specific epitope. In this case, epitopes of the spike protein of SARS-CoV-2. Now, with these protein antigens like SARS-CoV-2, the B lymphocyte needs to interact with T4 helper lymphocytes. And this is then going to allow them to be able to proliferate, differentiate, make different isotypes of antibodies, differentiate into plasma cells, do all the things they need to do to mount a strong immune response. So the T4 lymphocytes have also been activated by SARS-CoV-2 viral peptides on Mhc2 molecules on dendritic cells. And after these T4 cells proliferate, they differentiate into T4 helper cells, or T4 effector cells, that are going to make the cytokines that are going to promote immunity against the SARS-CoV-2 virus. Now, once the B lymphocytes have proliferated into a large clone of identical B lymphocytes, and once they differentiate into effector plasma cells that can secrete up to 2,000 antibody molecules a second, the antibodies that are made bind specifically to the S protein of SARS-CoV-2. And as we learned when we talked about defense against viruses, the binding of antibodies to this S protein neutralizes the virus by preventing it from binding to the ACE2 receptors on human cells. Remember, if the S protein can't bind to ACE2 receptors on our cells, it can't enter our cells. If it can't enter our cells, it can't proliferate and cause harm. So this is called viral neutralization, and we see this in the animation down below. So remember, in general, with the virus, the first step, as we learned in viral life cycles in Unit 4, is that the virus has to adsorb to receptors on the host cell. In this case, a spike protein on the envelope is binding to a receptor on a host cell. And that has to happen before the virus can enter and uncoat and replicate. So if we can make antibodies against these spike proteins ahead of time with our B lymphocytes and plasma cells, the antigen binding fragment, or FAB portion of the antibody, is tailor-made to fit these spike proteins. They cover up the surface of the virus, and that blocks the spike protein from binding to receptors on the host cell. And so the virus never binds, it never gets in to replicate, and the viruses become neutralized. Also, some of those can be opsonizing antibodies that can result in the virus being phagocytosed and killed by phagocytes. Now, in addition, when the T4 lymphocytes are activated by the dendritic cells in response to peptides from the SARS-CoV-2 spike protein, the primary role of the T4 cells, again, is to regulate immunity through the production of regulatory molecules called cytokines. So the naive T4 cells were activated by dendritic cells, recognizing peptide on Mhc2 molecules of the dendritic cell. And that allowed the T4 cells then to proliferate and differentiate into T4 effector cells that regulate the immune responses through the cytokines they produce. And so these cytokines can collectively activate or deactivate, enhance or suppress every aspect of both innate and adaptive immunity, depending on the population of T4 effector cell that's produced. Now, keep in mind, many of these cytokines produced by T4 effector cells promote inflammation, which is a critical part of our initial body defense against a virus. Remember, inflammation is how we initially deliver defense cells and defense chemicals to the infected site. And these cytokines are also, of course, critical for the activation, proliferation, and differentiation of naive B lymphocytes, T4 lymphocytes, and T8 lymphocytes, as well as other defense cells. So they're all very critical in the overall picture. But also remember that an excessive production of cytokines can lead to a cytokine storm, which can lead to serious and sometimes fatal COVID-19 infections, as we learned back under the SARS-CoV-2 life cycle. And that's when it becomes dysregulated. So the T4 lymphocytes are going to produce the cytokines that help the other immune cells function properly. And then, of course, the T8 lymphocytes, when they recognize peptide bound to MHC1 molecules, are going to proliferate and differentiate into cytotoxic T lymphocytes. We saw how they recognize it up above in Figure 3. And then once these cells differentiate into cytotoxic T lymphocytes, they'll use their T cell receptor and CD8 molecules to recognize SARS CoV-2 peptide epitopes on MHC molecules, MHC1 molecules on actual virus infected cells. And that then allows a cytotoxic T lymphocyte to kill the infected cell and stop them from further producing viruses. So again, what happens with the T8 cells is that the endogenous antigen, in this case it would be the spike protein, goes through proteasomes where the spike protein is chopped up into peptides. Here they enter the ER where they attach to MHC1 molecules. There they're transported to the Golgi and put on the surface of the infected cell. And then eventually cytotoxic T lymphocytes come along that have a T cell receptor CD8 that fit those peptides on those MHC1 molecules, bind and kill the cells. So here's an animation of a cytotoxic T lymphocyte recognizing peptide on MHC1 in a virus infected cell. So again, the infected cell chops up viral proteins into peptides, attaches them to MHC1 molecules, puts them on the surface of the cell, marking that cell for recognition by T8 lymphocytes. Remember, T8 lymphocytes only recognize peptide on MHC1. So here comes a cytotoxic T lymphocyte that has a T cell receptor and a CD8 molecule that fit that viral peptide on that MHC1. Now, once that binds, that triggers the release of granules, which contain perforins and granzymes. The perforins insert pores in the membrane of the infected cell, which allow these blue granzymes to enter. And these granzymes activate the caspase enzymes, the enzymes that are involved in apoptosis or cell suicide, which we'll see that. So that's inducing the virus-infected cell to die and break up into membrane-bound fragments, as we'll see in our next animation. So once these caspase enzymes have been activated by interaction with a cytotoxic T lymphocyte, that leads to apoptosis of that cell. And these caspase enzymes break down the nucleoprotein in the cell, and they also break down the cytoskeleton of that cell. And so the infected cell eventually breaks up into little membrane-bound fragments called apoptotic fragments. And these apoptotic fragments then can be cleaned up by other phagocytes called macrophages. So macrophages come along and engulf these apoptotic fragments and destroy all the remains, digest all the remains. If there's any viable viruses still inside, they would be killed too. And that's the apoptosis or programmed cell suicide, where the cell's cytoskeleton and nucleoprotein break down. The cell breaks up into membrane-bound fragments, which are then cleaned up by macrophages. And that's a way to get rid of cells that could be harmful to the body without inducing inflammation, which could prove harmful if there is too much inflammation, as we literally talked about cytotoxic T lymphocytes early in this unit. And during this whole process, memory cells are made. B memory cells, T4 memory cells, T8 memory cells. So if that same virus enters the body again, or if you get a booster shot at a later date, these memory cells will lead to anamnestic response. And that's where you have a heightened secondary immune response where more antibodies, more CTLs are made faster in greater amounts for a longer period of time. And typically that can then prevent symptomatic reinfections with that virus. And there we have the nucleic acid vaccines used for SARS CoV-2.